right, our second part on changing allele frequencies. The two things we're going to focus on here, actually it's one category, genetic drift, but there's two major types of genetic drift that we'll talk about, the founder effect and population bottlenecks. So when we talk about genetic drift, we're talking about fluctuation in allele frequencies from generation to generation that happen by chance. In other words, the allele frequencies change are occurring at random. That means that it's not affected by natural selection or anything like that. That's not what's causing them to change. It means it's just because of sampling size, their allele frequencies change by chance. Usually, it's when most noticeable if you have very small population sizes, and then that sampling changes the allele frequencies. Okay, if you have a really, really large population size, it doesn't seem to affect it as much. But when there's very, very small population, you can really see genetic drift happening more dramatically. So here you started out with an equal population again, and then just by chance, because of random sampling, these two categories seem to get a little bit larger, these two get smaller. Events that can create small populations that enhance genetic drift include migration, natural disasters, maybe a tsunami going through and wiping out a big chunk of a population, a geographical barrier that isolates a small group. Example, skin lightening condition called vitiligo, I think I pronounced that right, is more common in a small community in Romania that's isolated by mountains. So there they have a geographical barrier that kind of cuts them off from everybody else. Therefore, their sampling size is small, and that way if they're only around people that have that condition, and they're choosing to settle down and have children with people in that area, that gene pool is very concentrated. So the founder effect, this is when a small group leaves home to found a new settlement. The new colony may have a different allele frequency than what the original population has. What if the people who left just happen to be the few people that were carrying alleles for a certain genetic disorder, for example? Well, now if they're the ones that are leaving and starting a new settlement in a new area, most of the descendants that are going to result, the offspring and so on, that are going to result from that new population are going to have a very higher frequency of that genetic disorder, because we just happen to have those few individuals leave that had those alleles with them. So we have different founder populations that started out with only like a few people, 4,000 in Costa Rica, but after 12 generations, over 2,500,000. The Hutterites and their community, in the communities where they are located, 14 generations, now 36,000. So you can see you can start out with just a small number of a population that goes to a certain area, and then after many generations they can pass on all of those alleles that they have with them and really grow in population size. The Afrikaners of South Africa have a higher incidence of Porphyria variegata. So that's an example there of a founder effect. All individuals in a population with a certain illness have that same mutation. Like we already talked about the Bulgarian gypsies that have galactokinase deficiency. It can be evident in common illnesses where populations have different mutations in the same gene. So again, the Ashkenazi Jewish people In addition to having Tay-Sachs being more prevalent, a lot of them are more prevalent in having the BRCA1 gene that's associated with breast cancer. And so it's because these certain groups of people, as they broke off and founded in a new area, settled in a new area, they were carrying those alleles with them that coded for those different genetic disorders. It now became a higher prevalency in that population because they were marrying amongst themselves and having children together. Population bottlenecks occurs when a large population is drastically reduced in size. Remember, we said genetic drift doesn't tend to happen in large populations. So now what happens if you wipe out a lot of the population and you just have a small population size? Now you have what's called a population bottleneck, and the rebounds in the population size occur with the descendants of just a limited number of survivors. So whatever alleles those limited number of survivors had, that's what you got to work with for your gene pool. And you result in a very restricted, concentrated gene pool because you only have those few alleles compared to what the large population size originally had. Some examples of population bottlenecks. The Pingalapese people of Micronesia, say that 3 times fast, they had a bottleneck created by a typhoon that hit the island of Micronesia, and it killed all but 9 males and 10 females. Wow. Talk about wiping out almost the whole population. So their population had to rebound from just those few individuals. Because not only being geographically isolated, but also culturally isolated, that increased the frequency of a gene that caused blindness in them. They had a higher frequency of that because those alleles were concentrated. You've probably heard a lot in the news about the cheetahs in Africa, and this bottleneck was created by mass slaughtering by humans of the cheetahs, and also a change in their habitat after the most recent ice age, and so their population size really, really dwindled due to that environmental upheaval and just being hunted and slaughtered by humans. So you look at if they had 25 different alleles of a population, but now it got reduced to only these few, well, there's only so many combinations you can make with just those few genes. And so it becomes very, very reduced in terms of genetic diversity. Remember, we talked that genetic diversity is a good thing. That way, if a disease or something like that sweeps through, if you're genetically diverse, even if it kills off a little bit of the population, it won't wipe out the entire population, right? So you want to have genetic diversity. This kind of limitation can really put the cheetahs at risk for total extinction if there were to be a certain disease or something that was more lethal to individuals who had their limited, genotypes. Ashkenazi Jews, they not only have non-random mating because of, choosing to marry somebody and have children within their ethnic group or their religious group, but massacres over the years have caused just lower, those few survivors contributing to high incidence of certain genetic disorders, like we talked before, the Tay-Sachs disease. Remember, Jews were the ones targeted during, the time of Hitler, and as well as other massacres over the years. So that has greatly concentrated their gene pool as well. So these are some of the autosomal recessive genetic diseases prevalent among the Ashkenazi Jews. So I already talked about the Tay-Sachs disease, BRCA, the breast cancer gene, but a lot of these other ones too. Canavan disease, which is another. These are brain degeneration Brain degeneration. I mean, it causes early death. Death by age 4 for this one. I'm not sure in the Canavan, but I think that's really young as well. Niemann-Pick disease. Death by age 3. These are just terrible, terrible diseases. And so by concentrating the gene pool, so you have a higher chance of passing that on to your children. I mean, it's pretty devastating, which is why they developed a lot of, genetic counseling and stuff to very specifically talk to individuals who belong to this Jewish group to really help them decide, if they're going to have kids. Here's your chances if you're both carriers, or just to start that, just to check individuals to see if they were carriers, heterozygotes of the disorder, so that they'd be better informed to know what chances they would have of passing that on to their children. Okay, so genetic drift, which includes bottlenecks, population bottlenecks, and the founder effect.